So actually, thank you everyone who was able to come out tonight. I know it's the first in our fall series. We're all kind of getting back in gear. So thank you for making the time to come. I want to thank Dr. Lara-Gonzalez for joining us tonight, and just welcome him. It's in fact the first time that I'm meeting him, although it really shouldn't be, because he's got some definite overlap to the Stem Cell Research Center here. Really interesting. He's from Chile. I had a phenomenal postdoc and graduate student a few years ago who was a Fulbright scholar from Chile. I can speak firsthand to what an incredible educational program there is there. And what a great exchange it's been cross-border in that way. A lot of very well-known, very successful scientists. He received his PhD at the University of Manchester and did a postdoc at the Ludwig Cancer Center in San Diego, and UCI was lucky enough to capture him from there. Really interestingly for us in the Stem Cell Research Center, he works on the issue of how cells decide to divide. I'll call it that. Mm-hmm. Stem cells, obviously, this is such a critical part of what stem cells do in terms of the context of self-renewal, also really critical in terms of cancer. And as many of you have heard me harp on for some time over the last few years, really we need to focus on not just the late-stage translational studies or clinical trials, which of course we do in the Stem Cell Research Center also, but the very fundamental basic research that tells us how cells decide to do those things in the first place. And that's the intersection of research that we're going to hear about tonight. So it's my great pleasure to welcome him, both for the first time to UCI and this evening to the public lecture. And thanks very much.
Thank you very much. Can you hear me? Okay. So yeah, thank you so much for inviting me to present here. And yeah, I'm an assistant professor at the Department for Developmental Cell Biology here at UC Irvine, and I'm also affiliated with the Stem Cell Center. And so today I'm going to tell you about a story that we recently published on how cells control the amount of time that they spend in mitosis, and how worms can help us understand this process. Okay. So first, I'll tell you about a little bit about cell division. So I'll introduce some concepts of cell division, and also how cell division is affected by development and disease. I'll then tell you about our finding of what mechanisms ensure that cells divide at the right pace. And I'll finally tell you about our finding of how embryonic cells ensure accurate chromosome segregation. So as we know, cell division is very important for life, right? Each single one of us was born out of a single cell, the fertilized embryo that underwent many rounds of cell division until it became a fully formed human being. And so humans, us, were born with about 2 trillion cells. So that means that this cell has to undergo, or will have undergone, about trillions of cell divisions to give rise to our bodies, our organs, our cells. And not only that, but also we have that in our adult bodies. Divisions happen every single day, right? For instance, we have the epithelial cells, the blood cells that are constantly going under cell division. And so these divisions happen because stem cells in the body are constantly renewing and making more of these cells. But also we have other tissues, for instance, like the liver, that only regenerates under certain conditions. For instance, after a night of heavy drinking and your liver gets damaged, then these cells will be reactivated and start dividing again to make up for the lost cells. And so these cells are known as somatic cells, and it's important to understand that it's really critical that for each one of these divisions, that division happens accurately. So that means that there must be no errors in the process of division, because if there are errors, that can lead to diseases or conditions like, for instance, cancer. And so in my lab, we're really interested in understanding what mechanisms ensure that cells divide at the right time, at the right place, and also once they start division, how can they ensure that they divide accurately? So these are the main questions in my lab. So first, how do cells respond to external signals to decide to either divide or not divide? Then once they divide or they decide to enter into division, what mechanisms they use to ensure that division happens accurately? And finally, this is something new in my lab. We're trying to see whether we can exploit what we've learned about cell division to target cancer cells. And so today I'm going to tell you mostly about point number 2, what mechanisms cells utilize to ensure that they divide accurately. And so cell division happens during the stage known as mitosis. So here you can see the process of mitosis in a cell called RPE1, which is an epithelial cell. And as you can see, this is a very highly dynamic process. So you can see that the cell, for it to divide, it has to completely remodel itself. And that includes things like the nuclei, the plasma membrane, the cytoskeleton, the organelles. Everything has to remodel during this process of cell division. And so in my lab, or I should say here that also the important thing to pay attention to are the chromosomes that you can see here in magenta. So in my lab, we're trying to understand really how cells ensure that chromosomes segregate accurately during mitosis. So we all know that each one of us is composed of 23 pairs of chromosomes, right? 23 from our mothers, 22 from our fathers. And it's important that these chromosomes segregate accurately during mitosis, because these chromosomes contain all the genetic information that makes our cells. And so this is one of the main challenges of mitosis. How can cells ensure that they segregate their chromosomes in an accurate manner?
What does segregate mean?
Oh, split up in between the two different cells, the daughter cells. So when we talk about mitosis, right, we talk about the mother cell and then the daughter cells. And chromosome segregation refers to the process of these chromosomes being split up equally between the two daughter cells. Okay, so however, sometimes in mitosis there are errors that can happen, and here I'm showing you an example of such errors. So what we can see here is a cancer cell, a breast cancer cell called MCF7, and so you can see that in this case we have the chromosomes labeled in green, and you can see that this cell starts mitosis, right, aligns its chromosomes, but then what you can see is that when the chromosomes separate, there is one chromosome that remains right in the middle of these two masses. And so these chromosomes are known as lagging chromosomes, and what happens is that they often end up going to the wrong cell. So that means that one daughter cell will end up with more chromosomes than the other one. Or it can also happen that these chromosomes end up trapped in their own nuclei, and they're called micronuclei. So this is a very bad consequence for the cells, right? Because then that means that the information has not passed along accurately, and this is very typical in conditions like cancer. So this cell actually is known as chromosomally unstable, meaning that it often does this process of errors in chromosome segregation. And so we see, for instance, that in conditions like cancer, these errors happen very often. So here you can see chromosomes of a normal cell, where you can see that each one of these chromosomes is present in two different copies, right? One from the mother, one from the father. But in cancer cells, you often have these really bad chromosomal abnormalities. So you see, for instance, some chromosomes are present in 3 copies, others are present in 2 or 4 copies, and some are also rearranged at the bottom of this cartoon. And so these errors happen because cells make mistakes during mitosis, and these mistakes can actually help the cancer cells, because when the cells have these errors, they can evolve faster, and also they can resist to chemotherapy. Also, we have that in conditions such as, for instance, Down syndrome, we have that these individuals are born with 3 copies of chromosome 21, and these errors also originate not because of mitosis per se, but because of meiosis, which is a process similar to mitosis that originates the oocytes. And so in my lab, we're really trying to understand how do cells normally ensure that they segregate their chromosomes accurately, and then to understand the origin of these kinds of disorders or conditions like cancer and Down syndrome. And so in my lab, we use mostly this model system called C. elegans. That's short for Caenorhabditis elegans. And so C. elegans are these nematode worms that you find living in the soil or in the compost. And what they do is they feed off of bacteria that live in there. And so C. elegans is a very popular model system, and it's been around for many years. And people like using it because you can culture them very easily in the lab. You can also do the genetics with them. And one interesting thing is that they can go from one embryo to a fully formed adult in only 3 days. So that makes the genetics really easy, and they're really easy to manipulate in that sense. But in our case, we really are interested in using C. elegans because we find that most genes that participate in mitosis or in cell division are really highly conserved between C. elegans and humans. So that means that anything that we learn using C. elegans, we can directly apply to our own biology. And so what we do in the lab is mostly make mutants of C. elegans using technologies like, for instance, CRISPR-Cas9 that you may have heard of. And then what we do is we analyze these mutants using cell biology and also biochemistry techniques. And so here you can see in this cartoon, this is an adult C. elegans. And here inside you can see these are the little eggs that these worms produce. So C. elegans are actually hermaphroditic, meaning that they can self-fertilize, and each adult can make about 300 embryos each. And so most of the work that we do in the lab involves making these manipulations, and then isolating these embryos, and then filming them using microscopy. So here is a movie of a C. elegans embryo undergoing division. And again, you have the chromosomes in magenta and the microtubules in green. And in this case, what we use is mostly the first mitotic division that happens right after fertilization. So you can see that the cell first starts with these chromosomes condensing, then aligning in the middle of the cell, and then splitting up into the two daughter cells into a phase that is known as anaphase. And so the reason why we use these embryos is also because we find that these embryos divide very stereotypically, meaning that there's not much variation between embryo to embryo. And that makes it easy to, for instance, capture very small deviations from this pattern of division. And then we can trace back these deviations to specific pathways or specific genes. So most of the work that I'll tell you today about will be using these C. elegans embryos and trying to understand what mechanisms ensure their accurate division. But before I do that, I want to introduce another concept, which is time. So because time is really important for mitosis to happen accurately. And this is because during cell division, the chromosomes have to interact with these green structures that are known as the microtubules. And so during mitosis, the microtubules form a structure that is known as the mitotic spindle. And so chromosomes, they have to interact through these structures in pink here called kinetochores. And they have to interact with microtubules that emanate from opposite spindle poles. So the spindle is said to be bipolar because it has two poles that nucleate microtubules, and then these microtubule arrays go into the chromosomes and are able to be captured by the kinetochores. And so you can see here that in early mitosis, for instance, you have some chromosomes that have attached to microtubules and others that haven't yet fully attached. So these chromosomes that have attached are known to be bi-oriented, and these ones are mono-oriented. So what happens is that, for instance, these chromosomes are ready, but these ones are not. And so the cell has to wait before going into chromosome segregation, before going into anaphase, for all of the chromosomes to be attached. And so only when all of the chromosomes are attached or bi-oriented, then the cell can go into anaphase and can divide into two. So you can imagine, for instance, if this cell enters anaphase already at this stage, then the daughter cell here will receive 2 copies of that chromosome, and the one here will receive 3 copies of that chromosome. And that will result in aneuploidy.
So who's doing the counting? All of the chromosomes are ready.
Yes, so that's a great question. Who's doing the counting? So there are mechanisms. One of them, actually, the next slide will answer part of that question. But there are mechanisms that cell can use to make sure that all of the chromosomes have reached bi-orientation before it can go into the next phase. Any other question? Okay. So So we have here a situation, for instance, of a control cell. In this case, this is a cell that we use in the lab called U2OS. And you can see that this cell starts mitosis, then its chromosomes are aligning in the middle in metaphase, and then the chromosomes surrogate in anaphase. And this whole process took about an hour, a bit more than an hour. So we know there is a checkpoint, to answer your question, there is a checkpoint mechanism that ensures that cells have enough time. And this checkpoint really works by these chromosomes telling the cell, we're not ready yet, so give me more time. And then only then the cell can go into anaphase. And so what happens is that if we don't have these checkpoints, then this cell will go into anaphase prematurely. So in this case, within 20 minutes. And in doing so, you can see that there are massive errors with chromosome surrogation. So you can see that whereas in this case, the two masses look very similar in size and intensity, in this case, you have one mass of chromosomes being much larger than the other one. You also have a lot of chromosomes in the middle, a lot of lagging chromosomes, and this cell, both of these daughter cells will become highly aneuploid. And so I spent a lot of my career trying to study these checkpoints, looking at the mechanics. Oh, you have a question?
No,
Oh, okay.
I spent a lot of my career.
Yes, yes.
Don't tell everyone how long. But
Yes.
so how you label it? How you visualize it?
Yes. Yeah, so you can label the chromosomes in two ways. One way is by expressing these proteins, fluorescent proteins, like for instance GFP that you may have heard of. So these are proteins that normally fluoresce. And then if you fuse them to histones, which are proteins that bind to DNA, then you can basically have fluorescent chromosomes in your cell. In this case, actually, what we did was to use another technique, which is using a dye, so a compound that binds to the DNA, and when it binds, it fluoresces, and so then we can see the chromosomes only when, or the dye only when it's bound to the chromosomes. Yes?
My question was, what could cause something like a cell to not have a checkpoint, like an abnormality compared to the normal cell?
Yes. So there are mutations. So they're really rare mutations that can occur in cells that then will not allow the cell to have this checkpoint working. The reason why they're rare is because many mutations that affect this checkpoint pathway end up causing lethality in embryogenesis. So these individuals cannot even be born with these mutations. But there are conditions that cause mutations that are a little bit more mild. So one of them is called variegated aneuploidy, or no, MVA, mosaic variegated aneuploidy. And these are minor mutations in checkpoint proteins, and those still allow for these individuals to survive, but then they result in reduced checkpoint activity, and then these individuals usually have problems, for instance, with their development, and also with they have higher prone or tendencies to develop tumors. And but this is really rare. I think there's only been a few reported cases in the whole world, so it's really rare for this checkpoint not to be working in these cells. Yes. So as I was telling you, I spent a big chunk of my career trying to study the mechanics of this checkpoint and how the checkpoint proteins work together to sense these underlying chromosomes and to give cells enough time. But one thing that really motivated my future work is this observation that if you compare somatic cells versus embryonic cells, you see that the somatic cells take, as I said, about an hour to complete mitosis. But if you compare this to these embryonic cells, in this case, these are C. elegans embryos, we see that they only need about 4 minutes to complete mitosis. So they can do the same things that the somatic cells do, except that they do everything much, much faster. The other observation that we have is that these, compared to these somatic cells that need this checkpoint pathway to make sure that they have enough time to complete mitosis, these embryonic cells don't need these checkpoints. So you can inhibit the checkpoint in these cells, and you can see that the cells still take the same amount of time to complete mitosis, and in fact, they don't have any errors with chromosome surrogation. So then, from there, this is the question that we were trying to address here. First, what drives this embryonic mitosis to do division at such a fast pace? And the second one is, how do these divisions ensure their accuracy in terms of chromosome segregation? And so for that, what we did was use C. elegans. So we used this technique called RNAi, where you use double-stranded RNAs to target specific genes within the cells. And these genes, when they are targeted, they are depleted. And that allows us to evaluate what happens if you don't have specific genes in the cell. So what we do is we microinject C. elegans at this stage called L4 with double-stranded RNAs. We then let them recover between 36 to 48 hours at room temperature. And what we have is that these L4s then become adults. They start laying embryos. And what we do then is to take these embryos, mount them on slides, and then image them using this microscope. So this is the actual microscope in my lab that we use for these experiments, which is a spinning disk confocal microscope. And so what we do there is to make movies of these embryos, and then we can evaluate what happens to mitosis under these conditions. And so we've done this for many different genes in the cell, but one of the genes that we became interested in is called cyclin B3. And the reason why we use this or we study this gene is because we see that if you look at the control embryos, right, these cells start mitosis, then they do alignment really quickly, and then they do anaphase already in 200 seconds. However, if you don't have cyclin B3, you can see that these cells start mitosis, but then everything in mitosis becomes really, really slow. So you can see that chromosomes take much longer to form a line in the middle. The spindle here in green also takes much longer to assemble. And also, these cells take much longer to enter into anaphase. And so we quantify this. We measure the time from NEBD. So NEBD stands for nuclear envelope breakdown. That marks the beginning of mitosis, all the way to anaphase. And we see that in the absence of this protein, cyclin B3, we see that mitosis takes about 3 to 5 times longer. So that means that cyclin B3 is really important to drive the rapid pace of these embryonic divisions. Okay, so what's cyclin B3? So cyclin B3 is a protein that is part of this family of proteins called cyclin Bs, and they're called cyclins because their levels cycle up and down during cell division. So in particular, these cyclin Bs, they usually are low in interphase. So interphase is what we call anything, but that's not mitosis. So usually their levels are low. But then cyclin B levels rise right before the cells start to commit to mitosis. And cyclin B acts in mitosis as an activator of another protein called CDK1. And CDK1 is an enzyme, a kinase, that promotes protein phosphorylation. And it's the phosphorylation of these proteins that drives cells to go from interphase into mitosis. So then, at some point, cyclin B also degrades. And it's the degradation of cyclin B that makes cells go from mitosis back into interphase. So this is how it looks like in cells. So these are cells that were we labeled cyclin B with another protein called M-neon green. And you can see that cyclin B is only high, or it's only expressed in the cells that are about to commit to mitosis, like these ones. And you can also see that when cyclin B disappears, so it gets degraded, that's oops, it's a little slower, but OK. So that's about the time when cells finish mitosis. And you can also see, for instance, these cells that start with low cyclin B levels, but then cyclin B builds up until it reaches a maximum level that allows for these cells to start mitosis. So cyclin B is really important to drive mitosis, but one thing that we've known for many years is that cyclin B comes in 2 different flavors or isoforms that are known as cyclin B1 and cyclin B3. So most people think of cyclin B1 as the canonical cyclin, so the cyclin that drives mitosis in many somatic cells. But also we've known for many years that cyclin B3 is also there, it's also well conserved, and it's important in specific contexts, like for instance in embryos and also in meiosis. So meiosis is the division that gives rise to the gametes, or to, for instance, the oocytes and the sperm. And so cyclin B3 and cyclin B1 are both known to activate CDK1, so they both seem to be doing the same job. But as I told you before, right, if you don't have cyclin B3, then we see that the embryonic mitosis become very slow. So the next thing we ask is, okay, what happens to cyclin B1? Can it have the same phenotype as cyclin B3? And the answer is no. So we see again that compared to the controls, if we deplete cyclin B3, mitosis becomes very slow. But if we deplete cyclin B1, then it doesn't. And in fact, we see that anaphase in these cells happens slightly earlier than in the controls. And I'll get to that in the second part of my presentation. But this is suggesting that cyclin B3 is really important to accelerate the pace of these embryonic divisions. So next we asked, how can cyclin B3 drive this rapid mitosis? And so, as I told you, right, the main job of cyclin B is to activate CDK1. So then we asked, okay, is there a way, or is there a difference in the way in which cyclin B3 and cyclin B1 activates CDK1? And for that, we used these in vitro kinase assays. So what we did was to isolate either cyclin B1 or B3 from embryo extracts. We then incubated these cyclin Bs with histone H1, which is a substrate for cyclin B, and also radiolabeled ATP. So the idea is that if cyclin B and CDK1 have activity, then they will incorporate this radiolabeled ATP into the substrate, histone H1, and then we can see the radioactivity of histone H1 using a machine called phosphorimager. And we did this experiment in collaboration with the lab of Lee Bardwell here at UCI. And so what we found is that if you look at, so this is the radiolabeled H1, you can see that if we don't add any kinase, there's no radioactivity. If we add only cyclin B1, we see some activity, but if we add cyclin B3, we see much more activity. So when we quantify this, what we found is that the activity of cyclin B3 is about 25 times stronger than the activity of cyclin B1. And so we think that this could be explaining why cyclin B3 is really important to drive this fast-paced mitosis in these embryos, because it's a much better activator of the kinase CDK1. And so from here, we propose this model that cyclin B3 is acting kind of like the gas pedal of mitosis, so ensuring that mitosis happens at a really fast pace. And this is because cyclin B3 is a much better activator of the kinase that drives mitosis, which is CDK1. Now, if we don't have cyclin B3, cyclin B1 can still do the job of driving mitosis, but it's a much worse kinase, so it can only do slow mitosis. And so from here, we think that cyclin B3 is really key to drive the acceleration or the fast pace of these mitotic divisions, specifically these embryonic divisions, by hyperactivating CDK1. Okay, so now we think that we know why these embryonic divisions happen at a very fast pace compared to somatic cells because of the presence of cyclin B3 that drives the fast pace of this mitosis. But what about the checkpoints? The next question we wanted to know is if the checkpoint plays no role in ensuring the accuracy of this mitosis, then what else could be working here to promote this accurate chromosome segregation in the embryonic cells? And so to make a long story short, I'll tell you that the answer is cyclin B1. So as you can see here, we know that cyclin B1, when we don't have it there, we see that this mitosis still happen, but actually we found that these cells enter anaphase, so they enter chromosome segregation about 60 seconds before these controls. And so 6 seconds may not sound like a lot of time, but two things that we observed is that these divisions have higher rates of chromosome segregation errors, and also 6 seconds in this context is a lot, right? It's like 1/3 of the total mitotic timing. And so we think that cyclin B1 could be doing the job of the checkpoint in these cells by ensuring that cells have enough time to complete chromosome alignments and then ensure accurate chromosome segregation. Okay, so next we wanted to understand how can cyclin B1 be promoting or ensuring accurate chromosome segregation in these cells. So we looked more carefully at the mechanism by which cyclin B1 could be ensuring accurate chromosome segregation, and one of the key proteins in this process is called CDC20. So CDC20 is a protein that is an activator of another enzyme called the APCC. That stands for anaphase promoting complex / cyclosome. And this is a complex that drives cells to go from metaphase into anaphase by targeting proteins for degradation at the end of mitosis. So we know that the activity of CDC20 is very important for this process. So for instance, if we have CDC20 active too early, then anaphase will happen too early. If we now inhibit CDC20, anaphase will happen much later. And so we found this mechanism that these cells utilize to ensure accurate chromosome segregation, and it involves phosphorylation of CDC20 to prevent its premature activation. And so this is summarizing data from a paper from 2017 that we published, where we found that CDC20 is regulated by phosphorylation. So in early mitosis, CDC20 is phosphorylated, and we found that this is really important to keep cells in mitosis and to ensure that they don't go into anaphase too early. Then in late mitosis, CDC20 is dephosphorylated by the activity of proteins known as phosphatases that remove these phosphates and then activate CDC20, and then cells can now activate the APCC and go from metaphase into anaphase. And so in other words, we think that in early mitosis, the cells are in mitosis because of CDC20 phosphorylation, and in late mitosis, they are able to exit because of CDC20 dephosphorylation. And this mechanism would work in a manner that does not depend on the checkpoints. It would be something that these cells utilize to work around the checkpoints and to ensure accurate chromosome segregation. And so we also think that cyclin B1 would be working in this pathway. So we think that cyclin B1 somehow either promotes this phosphorylation or works together with this phosphorylation to ensure that the APC does not become active too early. And so just one piece of data. So we see that, again, if we don't have cyclin B1, anaphase happens too early, right? About 60 seconds before it should be. And also we see the same if we now prevent CDC20 phosphorylation. And in this case, we used a mutant that cannot be phosphorylated in cells. And so our proposed model is that cyclin B1 works to phosphorylate CDC20, and this gives cells about a 60-second delay to ensure that chromosomes align in metaphase before they go into anaphase. If we don't have CDC20 phosphorylation or we don't have cyclin B1, the effect is the same, and is that you have now cells entering into anaphase prematurely because they activate the enzyme, the APCC, too early, and this causes problems with chromosome segregation. And so our final model, so we think that cyclin B1 and B3 are working as the brake and the gas pedal of the car. So cyclin B3, as I told you before, accelerates mitosis, but cyclin B1 slows down mitosis or prevents premature anaphase. And it does so by working with CDC20 phosphorylation to make sure that these divisions happen fast, but not too fast. And so we're still working in the lab trying to understand how these two proteins work together to ensure accurate chromosome segregation. And also we're trying to understand whether this pathway contributes to divisions other than the embryonic cell divisions. And so finally, some take-home messages. So I showed you that embryonic and somatic cells use different mechanisms to ensure accurate chromosome segregation. We found that cyclin B3 accelerates the pace of these mitotic divisions, and also that cyclin B1 ensures that these mitotic divisions happen fast, but not too fast, by working together with CD20 phosphorylation to ensure that chromosome segregation happens accurately. And finally, worms are cool. So we've learned a lot about biology using worms. And I think it's really important to, as Eileen was saying, to consider model organisms. They can teach us a lot about biology. So finally, I would like to thank members in my lab who contributed to this work, especially Shabnam, Anne, and Amrutha. Also, Dr. Lee Balwer, who contributed to or helped us a lot with the kinase assays, my funding sources, and thank you for listening. Yes.
So your images are so beautiful, right? You can just so clearly see what you're illustrating. What's the advantage to a cell to go fast versus slow?
Yes,
Why
that's
do
a
cells want to do this?
right. Yeah, that's a great question. So why would cells want to go fast versus slow? So at the moment, we don't know, but we do know that if they go slow, for instance, in this case, if we either inhibit cyclin B3 or we do other perturbations that slow down mitosis, then that has consequences later in embryonic development. So these embryos are not able to develop properly and end up dying. So we're really interested to understand what's important about the pace of these mitosis. At the moment, we just know that it's important for embryonic viability, but we don't know exactly how.
Being on the subject that you described, the details of mechanisms, are we looking at concentration of respective CDK1 and 3 that determine the speed?
Yeah.
Or is it something else that comes into play, like available spaces where when the proteins can go in? Or is it something completely different that we don't know yet?
Yes. Yeah, so we initially thought that it could be concentration, right? So if cyclin B3 is more abundant than B1, maybe that's why it's able to drive faster mitosis than B1. So to answer that question, what we did is we used CRISPR to tag either B1 or B3 with a fluorescent protein, and we found that they are actually very similar in levels. So they're really comparable in terms of concentrations. And in terms of localization, we're also using these edited lines. We found that they are all, both of them are localized in similar structures. So they start initially in the nucleus, and then when cells enter mitosis, the nucleus actually breaks down and they become fully cytosolic. And then the only thing that we did find that's different is how they degrade, because at the end of mitosis, the cyclins degrade. So we found that B1 degrades first, and then B3 degrades later. So we don't understand why that's the case, or if it has any importance for cells to finish mitosis. But we don't think it's something to do with concentration or localization. But we are trying to explore whether we can, for instance, swap up different domains of the proteins because these proteins have structured domains that contribute to their activity. So we're trying to do experiments where we swap domains of these proteins and see whether that can, for instance, make B1 a better kinase or B3 a worse kinase. So we're currently working on that.
With your research and so forth, what are your expectations, and have you realized some of those expectations?
In terms of this particular research, you mean before going into this project, what were my expectations? Yes. Yeah, it's actually been an interesting ride because when we found first that cyclin B3 was slowing down mitosis, initially we thought, oh, maybe it's something to do with specific substrates, for instance, that B3 can phosphorylate some proteins that B1 cannot, and those could explain the phenotype. So it took some outsider's perspective to be able to realize that the reason why they're different is because their activity is different. And so this really told me that it's also important to communicate your science and have an outsider's perspective to look at your problem, because sometimes I think when you're too much in your own research and looking at your data too much, you sometimes can lose the bigger picture. And so it really took a collaborator to look at this data from outside to realize that this is because of the activity of the kinases and not something to do with, for instance, their preference for one substrate or the other.
Hi there. I had two questions. The first one was the relationship between cyclin B3 and cyclin B1. Is that unique to embryonic cells, or is it like all types of cells? And then my second question was like talking about the promotion of mitosis kind of made me think about like cancer, because in tumors there's that like kind of uncontrolled division. So I was just wondering if you had any like thing to add about that.
So they're both really great points. So it's been shown that B3 is expressed mostly in embryonic context and also in germline context. So it's not thought to be expressed outside of these two contexts. So I think that goes in line with the fact that somatic cells seem to be really reliant on B1, but B3 is basically because B3 is not there anymore. But talking about cancer, there are some kind of some sarcoma cells that do overexpress cyclin B3, and that was discovered less than 10 years ago, that there are sarcoma cells that have fusion proteins because of an X chromosome translocation, and in that case they overexpress cyclin B3. And so we're really trying to understand also whether the overexpression of cyclin B3 has any consequence in cancer, and whether it could drive cancer or not. So based on our data, for instance, we would anticipate that if you have expression of cyclin B3 in somatic cells, maybe that will drive faster mitosis or faster cell division. So we're really trying to address that question.
So I'll ask a question, then you're next. I'm struck, like, the embryonic stem cells divide so much faster than somatic cells. So does that have something to do with development and maturation? Is that clock the same across Drosophila and mammals, or is it different depending on species?
So we think it could be different between different species. One thing that we, when we were writing this paper, we were trying to find any data out there on mammalian cells and their mitotic timing, or vertebrate cells. And what we do know is that, for instance, in species like Drosophila and C. elegans here, the timing of mitosis is very fast. In other vertebrates, for instance, in zebrafish and in Xenopus, also the whole cell division is very fast. For instance, normal cell division is in vertebrates is like 24 hours. In these embryos, it's something closer to half an hour only. But we haven't seen any specific paper that looked at mitosis in particular. So, because it's really hard to do to label chromosomes in these vertebrate systems. Now, in terms of mice, I haven't seen any data measuring the timing of mitosis specifically. But I think it's something that I think we would love to do at some point, to look at this also in vertebrates.
Hi. Thanks for such an informative presentation. So I was just wondering, I remember you talked about a condition where CDC20 was not phosphorylated. In that condition, did you notice any errors in chromosome segregation in that particular condition? So we do notice an increase in rates of chromosome segregation.
an increase in the rate of chromosome segregation errors when we don't have this phosphorylation event. Having said that, we were surprised by the fact that these embryos still divided and they still are able to survive. So it seems like the rates of errors are low enough that the embryos can still survive and live with these errors. But we do see them, and in fact, you can push the system a little bit more by some perturbations that make the errors even worse. And in that case, the embryos die. But with low levels of chromosome segregation errors that we see with the lack of CDC20 phosphorylation, we see that the embryos are still able to survive.
Yeah, I have a few questions. So I'm not sure whether I got it. So you mainly your study is mainly on the embryonic cells, right? Not the, yeah. So, and also the concentration of the B1 and the B3 are the same in the concentration. So basically the activity is different. So my question is that what control them? So right now you see the increase of the B1 or B3 and there should be something like timer to control everything. So basically in the cell development, and the most important is what control it. This is something amazing. I am not sure. What do you have any idea about that?
So we see that one of the key mechanisms that can control this, or we think that it could be controlling, is what I mentioned about the phosphatases. So if I go back a little bit, so we think that this is the key because what we think is happening is that this phosphorylation is happening in early mitosis, but then how fast this phosphorylation goes away is what controls how much time the cells spend in mitosis. And so we think that that's the key, the actual phosphatases that remove this phosphorylation event. And we think that there are different populations of phosphatases that can respond to different things in the cell, like for instance, the status of chromosome alignments or something that could also be happening is that this is acting more like an autonomous clock. But we think that the phosphatases are the key for this process.
Any other questions? Ooh, one more. That was a great set of questions, guys.
Now that you mentioned photophase Ts, what is the mechanism that makes preference for that substance and not other substance to go in there? In other words, there has to be some sort of a key, electrical or electrochemical keyhole or something like that for that preference for that specific compound.
You mean this part here, right? The phosphorylation of CDC20? So we, at the moment we don't understand this part really well. And initially we thought that, maybe B1 is specifically required for this phosphorylation events in a manner that B3 is not. But at the moment, we don't have any evidence for that. So it could be that it's not really phosphorylation, the way these proteins are working together, but maybe something else. So maybe cyclin B1 is preventing dephosphorylation, or there could be other explanations out there. Maybe cyclin B1 is acting downstream of this phosphorylation. At the moment, we don't know exactly how these two are connected, but that's something that we are currently investigating.
Okay, thank our speaker for a wonderful presentation tonight. Thank you so much for coming out.